This video is going to explain how you complete the insect karyotype activity. In this activity, you are going to figure out how can karyotype analysis detect genetic disorders. So remember that karyotypes, this is just a picture, a visual representation of a living organism's chromosomes, and they are all lined up so that you can better analyze them. Introduction. There are two main types of chromosomes: autosomes, chromosomes that deal with traits other than sex, male or female, and sex chromosomes, chromosomes that determine sex. In order to view these chromosomes, a karyotype is made. A karyotype is a picture in which the chromosomes of a cell have been stained so that the banding pattern of the chromosomes is visible. The cells are then photographed through the microscope, and the photograph is enlarged. The chromosomes are cut from the photograph and arranged in pairs according to size, length, centromere position, and banding patterns. Karyotypes have become increasingly important to genetic counselors as disorders and diseases have been traced to specific visible abnormalities of the chromosomes. Objectives for this activity. Hypothesize how karyotype analysis can be used to detect genetic disorders. Construct karyotypes from the chromosomes of 6 fictitious bugs. Analyze karyotypes for chromosome abnormalities. Identify the genetic disorders of the insects by using their karyotypes. Assume that a new species of insect has been discovered. The insect has 3 pairs of chromosomes. Researchers have been able to trace 4 genetic disorders to specific chromosomal abnormalities in this insect. Study the karyotypes and phenotypes, which are the traits, of normal male and female insects as illustrated in Figure 1. So in this figure, you see 2 different karyotypes here. So on the top, you see a male bug with his chromosomes. So there are 3 pairs. You have pair 1, pair 2, and then that third pair are called the sex chromosomes, and that's what's determining whether this is a boy or a girl bug. So it says in the note here that normal male insects has a pair of sex chromosomes similar to those of the human male, one large and one small. In the same way, the female has a pair of sex chromosomes similar to those of the human female, both large. These sex chromosomes make up chromosomes pair number 3. So in humans, and this is the same case for these bugs, there are X and Y chromosomes. The X chromosomes are really big, so you see the male bug has 1 of those and the female has 2. And then the Y chromosome is much smaller, and the male has 1 of those Y chromosomes. So we say males are XY, and females with only 2 big X chromosomes, they are XX. So you will be using these chromosomes to figure out whether the bug is a boy or a girl, but you'll also be able to figure out if they have any chromosomal abnormalities to see if they have any disorders. So speaking of these disorders, the information below describes all 4 known types of disorders regarding your insects. For each disorder, there is a cause as well as a description of the characteristics that the insect will have, and a picture of what the insect looks like with that disorder. So in the table that you have in your document, you will see that there are 4 different disorders. So size reduction disorder, so they're just a smaller bug. Clear wing disorder, duplication disorder, unsegmented disorder, and then the next column in the table is telling you which chromosome is affected. So the size reduction disorder, this is an example of monosomy. So monosomy, that means that there's only 1 chromosome. So these bugs will actually be missing a chromosome. The clear wing disorder, this is an example of trisomy. So these bugs will actually have an extra chromosome. The duplication disorder, this is when a part of a chromosome is added onto the original chromosome. So this chromosome will be a little bit longer. And it looks like this is giving the bugs a double head, which is very interesting. And then the last one is unsegmented disorder, and this is a deletion of a part of one of the large sex chromosomes. So they're not missing a whole chromosome, they're just missing a part of one of them. So you will be referring back to this table in order to figure out which disorders, if any, your bugs do end up having. So in order to figure this out, which disorders your bugs have, you have to make a karyotype for each of these 6 insects. And they're all color-coded on here. So it looks like we have the first one is this teal blue color. Insect number 2 happens to be the yellow bug. But you have to arrange the karyotypes of these little insects. So you have to actually drag and move these chromosomes and line them up so that they look like the karyotypes that we saw previously at the top of your document, or like these, how we see right here. So you're going to use these figures as a reference, and you're going to determine the order of chromosomes for each of your insects. You will drag each chromosome into a horizontal line. Make sure the centromeres, which are that center part in the chromosome, make sure they're lined up to allow you to compare the chromosomes better. So we're going to do a little example together so you see how to do this. Here is an example bug. You don't have the red bug. This is an extra one. So for the red bug, this example insect, you need to use the reference images that you see on the right, and you have these in your document. But let's just focus on one pair of chromosomes at a time. So for pair number 1, this is the shorter, fatter sized chromosomes. And so when I look at my chromosomes that I'm given for this insect, I can see I have 1, 2 of those. I might not have 2. For your examples that you have to do, there might be only 1, there might be 3. It just depends if there's a mutation or not. But I happen to have 2. So I'm going to go ahead and move those and line them up just like that. So the goal is to get all of these red chromosomes in a nice straight line. Okay, next I want to look at pair number 2. These chromosomes are a little bit longer on the bottom and a little bit skinnier. So it looks like I have 1, 2 of those. I also have 2 of these. Good. So I'm going to move those and line those up next to the ones I already did. And then we move on to the sex chromosomes, which are the final pair, the third pair of chromosomes for these insects. And it looks like I have 1, 2 of those as well. Fantastic. So I am going to line those up. Notice how I'm getting the centromeres all perfectly lined up, or at least as good as I can make them. Okay, so that I can easily compare the lengths of all of these. So even though I do have 2 of all of them, some of the mutations involve slightly longer chromosomes or maybe slightly shorter chromosomes. And if I can line them up really, really well, it's going to be easier to spot those slight little differences in length of the chromosomes. But this looks pretty good. So I think I have finished this part of the activity. I've got my karyotype made. And so the next thing is that I'll be answering questions about my karyotype. So you will do this for all 6 of the bugs, and then you're going to pull up the questions on Schoology to answer questions about them. So you might have a question like, is this a boy or a girl bug? Well, it looks like I have 2 of the big X chromosomes, which are the sex chromosomes, the third pair there at the end. They're both big. They're the X chromosomes. If this is XX, that's going to be a girl. What disorder does this insect have? Well, when I look at these chromosomes, there's 2 of each of them, which is normal. They all appear to be the same length. None of them are missing small fragments or sections, and there's no extra pieces added on. So I'm going to say this bug is normal. It does not have any chromosomal mutations. It is perfectly normal. But what if it looked like this? What if the red bug would have looked like this? And it looks like there is an extra chromosome number 2. Well, we call that trisomy. Tri stands for 3. There's 3 chromosomes right here. That's a mutation of that chromosome number 2. And so when I'm answering my questions for this red bug that you don't have, it's just an example here, but I would make sure that I said This bug, the red bug, is a girl, has trisomy, which means she has an extra chromosome number 2. And then I would have to pull up this table here of the mutations, all the disorders they could have, and figure out which one she has. So trisomy of pair 2 looks like it's the second option, clear wing disorder. The extra chromosome of the second pair produces sterile, unable to reproduce insects that lack coloring in their wings. Since sterility always results, the clear wing disorder is not passed on to offspring. All right, so this bug would have clear wing disorder. So that's what I would answer for my red bug example. All right, so that concludes all of the directions for this bug activity. If you have any questions, please ask.